Our next speaker is Gideon Davies, a biblical figure from York University, who's going to talk like oxidasis. I've known Gideon for many years. When I first went to the synchrotron in Hamburg in 1990, that's when I met Gideon, who gave me a 5-minute lesson and said, okay, here's the book, use it. So, Gideon.
Thank you. Thank you very much, Joel. Thank you to the Israeli Academy and the Royal Society for being here, and thank you for Joe and the other organisers for making our life so easy since we've been here. It's a great honour to... Ooh, just press the right button. It's a great honour to be here. I'm going to talk today about some work on glycosidases. We'll sort of link through to sort of more medical aspects of glycosidases. And of course, as Joel pointed out, with a name like Gideon, it is great to be in the birthplace of the original and most famous Gideon. And for people who don't know what he did in the Bible, he led an Israelite army, but he was the first peer reviewer. And he actually didn't believe in God, and he set God increasingly difficult proofs involving more and more miraculous revisions in order to believe in him and go and lead an army. So it's a great pleasure to be here. A sad realization that I am in fact named after reviewer 3 is not great. But so I'm going to talk some work on glycosidase today, and it involves this particular lecture, 3 particular collaborations. A very long-standing one with Dave Cadlow, a brilliant scientist at Simon Fraser University in Vancouver, in Burnaby, who I first met when he was doing his PhD in Steve Withers' lab when I was on sabbatical there. Some probes, of which we had a little glimpse in the previous lecture, synthesized by Herman Overkleeft in Leiden, and a collaboration which I've not really talked about in public before with Israel Vladowski from the Technion, who's in the audience somewhere, and will answer all the hard questions at the end of the lecture for me. So carbohydrates. I'm here to talk about molecular fabric of life, to talk about carbohydrates. And I guess when I was a student, this is a slide I stole from Caroline Bertozzi, carbohydrates were considered the rather dull protective coating on the outside of cells, hiding all the really interesting biochemistry in the cytoplasm and the nucleus. And they were just some kind of protective coat. And studying carbohydrates was made even more difficult by the sort of chemical observation that they're the most diverse biological molecules. If you just take all 6, take a hexasaccharide of all known sugar monomers, there are, if I press the right button, there are 10 to the 12 possible isomers. So it means when you want to study carbohydrate active enzymes, access to substrates is very difficult. A synthetic chemist has to avoid making 10 to the 12 incorrect things, so synthetic chemistry is very hard. But it does mean that biology can use carbohydrate diversity as a language, the language of the cell, barcodes, postcodes on cells reporting on their physiological state, whether they're healthy, whether they're cancerous, whether they're inflamed, and so on. So there's a lot of biology that comes out of this carbohydrate diversity. So we do now know that organisms, and my carbohydrates are just wiggly lines, wiggly lines here, that we have glycosylation inside cells, and I'll talk a little bit about that, but the cell surface is covered in glycans, which are involved in communication with foes, such as bacteria and virus, think of the flu virus, for example, and communication between healthy cells, or between killer cells and cancer cells. These are all glycan-mediated, so there's a lot of biology there to study. And of course, one of the great leaders in that field was a great Weizmann scientist, Nathan Sharon, who sadly passed away 9 years ago, who of course described the study of glycans a little bit provocatively as the last frontier of molecular and cell biology. So that's what I'll be talking about today. But since I was young, glycans are now seen a lot more in medicine. For instance, we all know about the anti-flu drugs that are glycosidase inhibitors, Tamiflu and Relenza. Our children, not people of my age, but my children will have been vaccinated with carbohydrate vaccines against bacterial capsular proteins. We see glycosidase enzymes in the clinic now for enzyme replacement therapy, including Cerezyme, famously solved by our chairman here. And coming through clinical trial, did very well in phase 2, less well in phase 3. The first glycomimetics here to prevent vascular crises in sickle cell patients, actually blocking glycan decoders on cell surfaces. And perhaps some of the most exciting work is in the link of tumour cells and killer cells in cancer and the development of cancer-specific glycan antibodies as anti-cancer things. So there's a lot of glycobiology slowly entering the clinic, and I thought I'd talk about some examples of that from our own work. So I'm going to talk today about glycosidases. Enzymes, there's going to be a little bit of chemistry in my talk, sorry. Enzymes that break glycosidic bonds. And as we saw in the last lecture, we've got 2 catalytic residues. We've got a nucleophile in this case, which is going to make a nucleophilic attack. And we've got an acid in the Bronsted sense, going to protonate this leaving group, otherwise a poor leaving group, to allow it to leave. And then in a second chemical step, for these enzymes whose product has the same configuration as the starting material, a water comes in, or something else. In a second step, double displacement, and we get our product. And these enzymes have phenomenal rate enhancements. If you compare the catalytic rate, kcat, with the uncatalyzed rate, it's about a 10 to the 17-fold rate enhancement. And that has a second implication that Wolfenden pointed out. It means there's phenomenally tight binding of the transition state. And that means if you can make transition state-based mimics, you've potentially got very tight binding compounds if you can somehow mimic some of these reactive transition states. So I'm going to take 2 examples today, both one inside and one outside the cell. I'm going to start by looking at a little bit of historical work, but it's now in the clinic, on the O-glucnag modification, this intracellular glycosylation with a single sugar that's reciprocal to phosphorylation. And then in the second half, I'm going to talk about my work in collaboration with Israel's group on the degradation of these glycosaminoglycans, particular heparan sulfate, and its link to cancer and anti-cancer compounds in breast cancer models. So I'm going to go through these 2 examples. So O-glucnag, this is a sort of the famous intracellular glycosylation. It's a single sugar, N-acetyl glucosamine, on serine and threonine residues on intracellular, it's cytoplasmic and nuclear proteins. Okay, now glycobiologists, they don't like showing chemical structures, so sometimes I'll show it as the glyco blue square. But more than off, I'll inflict the chemistry on you. And this is massively widespread. You know, most of your proteins inside cells have O-glucnag. It's you know, here's an antibody blot from a paper by Lance Wells in Science a little while back. And it's a dynamic glycosylation. It's not like other glycosylations where you put the glycan on and that's it until the protein's degraded. This is a dynamic modification. It's going on and off many, many times in the lifetime of the protein. And it's largely reciprocal to phosphorylation. So it is the same serine and threonine hydroxyls that are receiving O-glucnag as are being phosphorylated and dephosphorylated by kinases and phosphatases. But in contrast to this huge array, and these numbers are much greater now than on my slide, of kinases and phosphatases, this is not so subtle. There is one enzyme installing this sugar inside your cells, and there is one enzyme only, called the O-glucnag hydrolase, historically HEC-C, which is taking that sugar off. And it's clearly far less subtle, okay? We've not got exquisite signaling cascades here, because we've only got 2 enzymes involved. But the implications for much of O-glucnag biology is because of this link to phosphorylation. If you have O-glucnag, and its level changes with diet, if you had a couple of those cakes on the table, your O-glucnag levels are far higher than people who just had a glass of water, okay, seriously, you can't have a lot of these events taking place whilst there's still a sugar. So it's a very complex dynamic equilibrium between whether you've got O-glucnag, hydroxyl, or phosphate. So we'll just go through these 2 enzymes. I shan't say too much about this one, but the O-glucnag transferase, only one in humans. It's taking an activated sugar with a good biology's leaving group, UDP, and we get a nucleophilic attack here to make our beta-linked glucnag. It's a really interesting protein, but not one for this lecture. It has about 13.5 of these TPR repeats, first solved by Elena Conti's group. And then it has a catalytic glycyl transferase domain. There's a few TPRs here, first solved by Suzanne Walker's group back in 2001. About a week before Dave Barford interviewed me for a fellowship to work on the structure of that, rather unfortunately. But anyway, and this is binding many, many protein partners and somehow dictating specificity. It's a huge area for future research. And in order to try and access the full-length structure of those, we have sort of moved very recently into Cryo-EM. So here's I'm very proud, our first ever Cryo-EM data from a few weeks ago, showing a sort of dimer arrangement of these things. And that's our sort of almost full-length bit of disorder up here, Cryo-EM structure of the O-gluconate transferase. So seeing how this will interact with ligands and other protein partners is a big work for the future. So I'm going to talk today about inhibitors of the hydrolase. Okay, I want to go through some of this work here. That back in 2004, O-gluconate was found on the tau protein, which we heard a lot about yesterday. Around about 2006, the first structures of bacterial homologs came out, and particularly this mechanistic analysis here, which I'll go through in a couple of slides. The structures and the mechanism together gave the first generation of inhibitors of this enzyme, which could be used to probe the cell biology of O-gluconate, and which could go into animal models, initially healthy rat models, to see if you could perturb O-gluconate in animal models, and then subsequently into humanized tau and amyloid mice models to actually study whether you could influence neurodegeneration and the progression of disease. And somewhere around about here, Electos, a spin-out company from Simon Fraser University, and Merck came on board, and just before Christmas, they announced what their phase 1 clinical candidate against this target was. So I think we'd sort of go through this evolution of work here. So Gong's lab, back in 2004, pointed out that tau contains a number of O-gluconate sites. And it's been known for a long time that disease form of tau, if you look at post-mortem brains, is hyperphosphorylated. And what Gong's lab subsequently showed was that if you look at post-mortem brains, indeed they've got about 4 or 5 times more phosphate, but they've got about 4 or 5 times less O-gluconate on tau, at least suggesting that this reciprocity has disease implication. And this paper here was the first one where indeed people could knock out the O-gluconate hydrolase with a very promiscuous inhibitor that no one uses anymore, with the idea of shifting this equilibrium over here. So when we sort of started this project round about the sort of early 2000s, we were interested in looking at the mechanism. I'm interested in reaction mechanism and curly arrows and how that might lead us to design better inhibitors. And so we're going to have a little bit of chemistry. So here we have a beta-linked sugar. The key thing about this enzyme is when it performs hydrolysis, the product has the same configuration at the reactive carbon as the starting material. That's retention of configuration in chemical speak. Now, I've shown you how that reaction is normally done. Okay? It's normally a 2-step reaction. A nucleophile attacks on the enzyme. It kicks out a leaving group. And in a second step, a water comes in. 2-step reaction gives net retention. But chemists amongst you know, when you've got an acetyl group at the 2 position, another option becomes available called neighbouring group participation. So which of these 2 mechanisms is it? Because if you're going to make a mimic of this reaction, you need to work out which of these 2 mechanisms is. So could it be neighbouring group participation, where instead of a nucleophile on the enzyme, you use your built-in nucleophile at the 2 position of your sugar? This nucleophile can make a nucleophilic attack, kick out the leaving group, and you get this bicyclic oxazolene intermediate. So which of these 2 mechanisms was used by the O-gluconate hydrolase? And here we turn to some classic physical organic chemistry. We turn to fluorine. Okay, the most electronegative of the elements. It's pulling electrons out of the system. So if we've got this sort of mechanism, the more fluorines we add on here, the more electrons come out of the system, the less nucleophilic the oxygen is, and the reaction rate should go down. And that's exactly what is observed. Each successive fluorine massively reduces the reaction rate. O-gluconate hydrolase uses neighboring group participation. And as a structural biologist, you can play with some of these compounds and find one that has just the right rate constant that you can squirt it in a crystal, freeze it, mount it without it reacting, and that's what we did. If we go over to this thing here, Yuan here, PhD student of, oh, how do I get over there? Oh, there it is. Ooh. Ooh, yeah, it's quite hard to get there. So, taking advantage of this Taft analysis with 2 fluorines here, Yuan he was able to track this distorted Michaelis complex of the O-gluconate hydrolase, where indeed the N-acetyl group is now in perfect position for nucleophilic attack. And that then goes on to react to form a stable, on a mutant enzyme at least, a stable bicyclic oxazoline. So by taking advantage of this chemistry, we could understand the mechanism and also use that to inform structural biology to trap this enzyme at different points along its reaction coordinate. But these oxazoline intermediates are rather lovely. When you've got this kind of mechanism, put distorted sugar here to allow an inline attack with this intermediate, and there's the structure of the intermediate. A long time ago, Spencer Knapp in the States had proposed and shown that a mimic of this with a longer carbon-sulfur bond, more transition state-like, would be a powerful enzyme inhibitor for this class of enzyme. And indeed, thiazolines, and here's the structure of, oh, that's an oxazoline, on the enzyme, were pretty good 70 nanomolar O-gluconate hydrolase inhibitors. But 2 or 3 other enzymes in cells, notably hexosaminidase A and B, the Tay-Sachs enzyme, also use that mechanism. And so getting an inhibitor that would inhibit the O-gluconate hydrolase for cellular studies, but not give you Tay-Sachs disease symptoms, was the focus of sort of structural-based work in future. And we were fortunate when we solved the first structure of a bacterial homologue, whose active site was very like the human, a massive cavity underneath the N-acyl position. So over several years, and this one slide probably has about 10 years' work on it, we were able to sort of go from these mid-range nanomolar thiazolines with no specificity through a series of compounds exploiting this space in structure through to something with sort of mid-range picomolar binding specificity, which didn't inhibit the human hex A and hex B to any extent. More subsequently, the compound announced by Merck and Electos just before Christmas has better pharmacokinetics by incorporation of these 2 fluorine groups here. And that's the compound that's in clinical trial. But all of this was on bacterial homologue structures, which is very similar in this area of the structure, but down here it's quite different. So with the knowledge that these things, might be going through to clinical trial, we set about solving the human structure, which is a much more complicated structure. And here I feel embarrassed. I've gone to all these lectures at this meeting about the importance of disordered regions. We spent 10 years trying to cut off disordered regions from our protein structure. I'm trying not to look at the front row here, because I'm going to feel quite guilty. So I'm going to carry on looking up there. And it was known in this paper, a little throwaway line in this paper, is that there's a caspase cleavage site in the O-gluconate hydrolase. It's a complex protein. It's got a catalytic domain, an N-terminally disordered region, a large disordered region here, some helix bundle here, and a domain with sequence similarity to acetyl transferases, function still unknown. But it was known that caspase could cleave here, and that if you separated it, the 2 halves reassemble to form a functional enzyme. And that led a number of labs to come up with strategies whereby you co-express this ordered helical bundle with varying lengths of disorder domain here, and the catalytic domain, again, with varying lengths at each end. And you see the results. You can express the catalytic domain and the helical bundle, on the slide that works. But that, like I said, that was probably 10 or 15 postdoc years of work changing domain boundaries in my group. It's probably the slowest thing we've ever worked on, with the most soul-destroying work that I imposed on my group. And when we finally solved the structure, a number of other groups had come up with that strategy, and indeed 3 structures all came along at once, 2 in Nature Chemical Biology and 1 in Nature Structural Biology, and all reporting similar approaches to isolate helical domains and catalytic domains and have them reassemble either through co-expression or making sort of permuted enzymes and so on. So you've got a human catalytic domain structure, which is now allowing the next generation of inhibitors, which I'm not going to talk about in the 2 slides, to be developed. The original thiazolines, but also now moving into those parts of the structure which are human-specific to try and get even better inhibitors. And there's a lot of inhibitors now around the low nanomolar, picomolar level being designed to exploit some of these extra interactions. So is any of this any use? Well, yes, the hallmark of neurodegenerative disease of tau is hyperphosphorylation of tau. So could we knock out this O-gluconate hydrolase and hope that if the transferase continues to act, we would drag this equilibrium over towards a higher O-gluconate state, and therefore perhaps reduce the toxic levels of tau in cells? And I'll put all my results, and that a lot of results of David Cadlow's group from this second paper on this one slide, but that has worked very well. On healthy rat models, you can go in with these specific thiazolines, chain extended to be specific for that binding pocket, and indeed increase O-gluconate levels and decrease the pathological priming sites on tau. Okay? And in work that is Dave's and has nothing to do with me, he then moved into the frontotemporal dementia on chromosome 17 humanized tau mice, which are a great model for early onset tauopathies. And indeed, on these humanized tau mice, you see major reductions in hyperphosphorylated tau for the mice fed on the inhibitors, and likewise in soluble tau. And in these sort of neuronal counting experiments, you indeed see these mice have more live neurons after the application of these mechanism and structure-based inhibitors. So that's pretty exciting, and that is, what led pharma to get it interested in this sort of work. But these observations may not simply be limited to tau. Okay, there's been other work from other groups. This is not my work. Matthew Pratt's work showing that if you, and this had to be done chemically, but if you O-gluconagolate the single O-gluconate site on alpha-synuclein, it prevents aggregation. And this is around about residue 80 in the human. It's in your necessary and sufficient, that was neither necessary nor sufficient region, and prevents these fibers being formed from alpha-synuclein. And perhaps my favorite experiment from David Cadlow's group is this one here. This is the Morris water maze, where the best model, a model, for dementia, which is you drop your mice into their swimming pool, and there was visual cues around the room, and they swim around, and somewhere there's a perspex hidden feeding platform, and after some training, the mice learn where the feeding rescue platform is, and they swim straight to the platform. The amyloid, the various neurodegenerative mice can't do that. They don't remember where the feeding platform is, and they just randomly swim around the pool and don't learn anything. The key thing here is these double amyloid tau mice on the thiazolines behave like wild-type mice, and they can remember where the feeding platform is. So although I didn't do that work, it's really lovely as a structural and chemical chemist, to see that an idea which started with trying to work out a reaction mechanism, measuring rate constants in a spectrophotometer, has eventually led to compounds where mouse models don't show so much cognitive decline. So that's pretty exciting. So that's sort of one example of the sort of work we do in my group, trying to study enzyme mechanism and build on that to get compounds that can be used, hopefully, clinically. And that brings me to my collaboration with Israel at the back there, for which we're going to move to a far more complicated polysaccharide, heparan sulfate, polysaccharide with many chemical modifications, with sulfation, etc., etc., and its role in cancer progression. And there is a massive, incredible body of work from the emotional and intellectual father figure of the field who's sat at the back here. And we were particularly interested in heparinase, the enzymes that breaks heparan sulfate. So let's just have a look what that enzyme does. We've got quite a complicated polymer here. I've got my glyco language here, some chemistry here. It cuts after glucuronic acid, but only on quite a complex heparan sulfate. We've got different sugars. We've got glucnag here. We've got sulfated glucosamines here, O-sulfated, N-sulfated, quite a complicated substrate to be working on. We've got iduronic acid, which has been epimerized at the 5 position, a very complex thing to work on. But the role of heparinase, especially has many roles, but in the cancer context, it's being secreted and it's degrading these extracellular matrix glycosaminoglycans. Overexpressed in cancer cells, degrading these glycans, not only making cell proliferation easier, but releasing all the growth factors which are stored in this area to allow even more proliferation. So there's a number of compounds already in clinical trial, phases 1 to 3, on heparinase. So we've got a number of different compounds. We'll look at the structures of the top 2 against myeloma, breast cancer, pancreatic cancer, and so on and so forth. But these are, any medicinal chemists in the audience are going to squirm here. These are rather complex. They don't look like drugs you'd want to be taking. Massively complex mixtures of polysaccharides with random degrees of sulfation, random chain lengths, 20 to 22 sugar units here, 6 to 8 repeats of the whole thing for SST001, which is probably one of the best compounds out there. PI88, I don't even know how this works. This is an alpha mannose polymer that's sulfated and phosphorylated. Compounds, when you look at them, you're thinking they're going to bind to a lot of things inside human cells or inside organisms, and yeah, they don't look like drugs, do they? So we're going to get to the point of trying to design better compounds for these analyses. But for the young people in the audience, I'm going to admit, the old Glucnag project, we knew at the start where we wanted to be at the end. This project, completely serendipitous. It started with a totally different project. We were working with Herman Overkleeft, and you've heard about some of his compounds before, on probes to image enzymes involved in lysosomal storage disease as diagnosis. Tracy Gloucester, in my group a long while back, had worked on some of these beta-glucosidases that use this attack with a nucleophile on the enzyme in this case, giving rise to this covalent species. And she'd worked on cyclophilitols, involving a new synthesis by Robert Madsen, related but more specific than some historical compounds in the literature. And you can either have an oxygen here for cyclophilitol, or in subsequent iterations, a nitrogen making an aziridine. These are really reactive, irreversible covalent inactivators of enzymes. And you can either hang fluorescent groups off on this Y position here, or on Z. We saw examples of those in Sal's talk. Or you can hang them off the aziridine nitrogen here. And these can be fluorescent groups, biotin for capture, which are used in the biomass applications, but mainly fluorescence. And we've been working on these with Herman Overkleeft as diagnostic tools for lysosomal storage disease. So the beta-gluc version with a substituent here can diagnose healthy adults and various Gaucher patients with different mutations, with different severities, or people who are receiving the enzyme replacement therapy Cerezyme. Likewise, alpha-configured ones work for Pompe disease, the galacto for Fabry disease. And again, you can get diagnosis here of child and adult Pompe patients. So that's where we were and how our mind was working. And we wanted a compound that would look at human beta-glucuronidase, the GUS-B enzyme, for which there is a lysosomal storage disease. And so Hermann Overkleeft responded to our request and made these aziridine glucuronide probes. And we used those on one of my postdocs' platelets. So I literally did get blood out of one of my postdocs. So this is Liangru's platelets. And not only did this probe inhibit and label the GUS-B, that's these bands up here, it gave fantastic labeling of human heparinase. Now, human heparinase, I've said, has a long active site, as we'll see, demands a really long substrate, complex sugars, various degrees of sulfation. But this probe, and probably the only time in my career where a monosaccharide probe has inhibited an enzyme with a large active site that normally leaves 5, 6 sugars, fantastic labeling of human heparinase. And with that observation, that led us to the project I'll just go through some slides on now. So human heparinase is quite a complicated enzyme. So, well, before we get to that, so this here, I ran into Liangru's office, I said, wow, that's fantastic. And he said, it's heparinase. I said, we stop everything and we work on this now, because this is a really important enzyme. And he gave me that rather cynical look and then leapt in with great enthusiasm and was absolutely brilliant. So heparinase has always been a challenge for structural biologists. I know a number of people here worked on it. It's synthesized as a sort of pro-enzyme, and there's a cathepsin L cleavage which cleaves off a loop in the protein structure to give the mature active heparinase. So that was always a challenge to get enough material to do structural biology, but back in 2003, the McKenzie group had shown in baculovirus systems, you could actually do like we did for the human O-glucokinase, do a dual expression of one half of the protein and the remaining 8 kilodalton fragment, and these reassembled to form active heparinase. And so that's the strategy we took to get baculovirus expression. It worked. After a lot of work optimizing this to optimize the expression levels that Liang did, we were able to get a 3-dimensional structure of the human heparinase as its active enzyme form. So there's the structure. There's a Timbarrel domain, nothing special there, little beta sheet domain here. And then we were able to try and probe the enzyme subsites and see what subsites require what ligands, which needs sulfation. Of course, then you do hit the chemical challenge I mentioned. You simply don't have access to most of these substrates. So we did a really intellectually challenging experiment. We bought every single substrate you could possibly buy and soaked them into crystals over a period of months and used a lot of synchrotron time to try and map through the active site what... The hard thing here is it appears on that screen, but not on mine. Oh, there we go. To try and map through the active site what subsites would bind particular sugars. So we can zoom into the enzyme active site, and these are commercial ligands that were available. I think this one from Sigma. It's unsulfated, and you can zoom in and see the interactions. Here's glucuronic acid. It's actually got a good leaving group, but isn't a substrate. And you can then go for various sulfated things and begin to map a functional group map down the active site, which sites require sulfation. We've been trying to use that information to design better substrates for the enzyme. This is some unpublished work with Vito Ferro, trying to get a coloured substrate for heparinase, because that is a big challenge. If you want to go into drug screening regimes, you really need something coloured. And so we worked with him on on a fluorescent disaccharide that's appropriately sulfated. And it is a substrate, but it's still not a very good substrate. But it did allow us, with quick soaks, to access a nice, well, that's the original unsulfated ligand that wasn't a substrate, but bound. Here is the methyl umbelliferol, fluorescent leaving group for high throughput screening. Nice distorted sugar here, and a bisulfated ligand up here. And we can see that beautiful inline nucleophilic attack for us to make an attack here and kick out this leaving group. And this substrate works, but probably still not good enough to go forward with other strategies. But in order to try and do some kind of drug screening to develop new inhibitors or screen compound libraries, this compound so far hasn't been useful, but the fluorescent probes have been fantastic for that. So, I think we'll stop you. That's just going to irritate everybody, isn't it? Right. So these overclafed, designed and synthesized aziridines bind to the enzyme. They're distorted into this reactive conformer. There's nucleophilic attack. This is an inactive variant here. And they go on to form these covalent adducts. And like I said, we can hang fluorescent groups or biotin off the nitrogen and start to think about drug screening regimes. These compounds work in, these fluorescent compounds work in human cells. They can image both heteranase and actually the proform of heteranase, whose structure we subsequently solved, but I'm not going to talk about that today. So here's some purified proteins. So we get good labeling of the proenzyme, so we can actually track this process, and good labeling of heteranase itself. And this, the power of having a sort of an activity-based probe like this is that you can now start to do all sorts of biochemistry on heteranase that wasn't previously accessible. Wow. Brought the weather from England. And one of the many things we use it for, and here I'll show it a gel assay, is to screen libraries or assess the potency of potential inhibitors. So you can do competition assays. So here's our probe, which you know about, and here are 3 potential drugs whose structures I'm not going to disclose. This first compound here, so what we're doing here, and to help the Hebrew people in the audience, I'm going from left to right with my concentrations. Oh no, that's right to left, sorry. Right to left with my concentration. So as we increase the concentration of this putative inhibitor here, you'll see we compete with the binding of this probe for the human GUS-B enzyme, and we can measure binding constants. This particular compound doesn't inhibit human heteranase, so we increase the concentration and we still get lots of labeling with this probe. And we can go on and screen different compounds. Here, a different compound. It's a really good inhibitor of human heteranase. You can see the amount of labeling from the fluorescent probe going down. But it doesn't inhibit, to any reasonable extent, the other human glucuronidase. So you can begin to look at specificity of enzymes and begin to find we've got some really nice tight-bounding sort of nanomolar IC50 compounds here that are really good inhibitors of human heteranase. And a gel is very easy for us to see in a lecture, but these compounds are also really amenable to doing fluorescence polarization assays, so you can upscale to 96 or 384 well plates and start to screen much larger compound libraries to get heteranase inhibitors. So that's a really exciting development of where we're going with these probes. So have these probes, have these inhibitors that we've discovered through these processes, now we've got access to probe, been any use? So this is work Liang did in Israel's lab on a short-term EMBO scholarship. Yes, sort of standard way of looking at heteranase inhibition is to start with some assays looking at the degradation of heteran sulfate on these extracellular matrix layers and actually see how much solubilized heteran sulfate you can produce. And indeed, these inhibitors are very good at inhibiting the enzyme and preventing the degradation of human heteran sulfate. The next step in these experiments is to go into a sort of metastasis and cell migration assay using Matrigel, where you have a gel which has heteran sulfate proteoglycans, and you're measuring to see if cells can dissolve that matrix and migrate through and then measuring how many cells come out the other side. This is a really good cell proliferation assay. And again, the two of the compounds we saw on the previous slide work really well to prevent this proliferation and migration of cancer cells using this glioblastoma model. And I guess most recently we got some fantastic results looking on Israel's breast cancer models with some of our compounds. And here we're looking at a fluorescent breast cancer model, and you can see that one of our compounds, compared to sort of the current best SST0001, getting very similar reductions in breast cancer based on compounds which we've been able to access because of the structure and the activity probes together. And so that's where we are now. We're going to try and improve these compounds, exploit what we know about other parts of the active site to see if we can get even better compounds. But these are looking, as promising as the phase 3 clinical trial compounds, but they are small molecules with more drug-like properties. So I'll sort of just finish talking about carbohydrates and the molecular structure of life. I like to abuse retrosynthetic arrows here. But sort of our strategy in the lab is to try and understand enzyme mechanism in light of three-dimensional structure. And one project, we were looking at imaging enzymes, and that has indeed led to anti-cancer compounds. And certainly understanding this mechanism and structure of the human O-glucnag hydrolase has led to mechanism and structure-based compounds which work in cells and in animal models for tau. So just some aspects of glycosidases, and you've seen some compounds in clinical trial, and hopefully there'll be a few more. So I'd just like to thank everybody, our 3 collaborators, which I mentioned at the beginning, the people in my group, Zach and Liang for lots of the heparinase work, Richard and Jamie for the cryo-EM, and a lot of the O-glucnag work, which was a long struggle of Christian Roth, Alexander Males and Sherry Chan, and Imogen Breen for some of the Gaucher work, and the funding bodies, and certainly the UK synchrotron facility, without whose fantastic facilities we would never have done any of this work. Thank you very much.
Great. So thank you, Gideon, very much. I think it's a pleasure to hear how chemistry still lives and can influence structural biology. Are there questions? Xiaodong, please.
Just more general question. Why do you think so many, there are so many kinases, phosphatases, but only one enzyme of your interest?
Oh gosh, there's the billion-dollar question in the O-glucnag field. Absolutely. It does have specificity. The transrase is influenced by other binding partners to the transrase. And a lot of people's work suggesting actually it can also recognize areas of differential phosphorylation. So there are elements of control, but we simply don't know them. I mean, clearly it is not there as an intimate signaling cascade. It's much more of a tidal wave going through the cell in response to diet, but also in response to stress. All sorts of forms of stress, you'll see O-glucnag levels increase. So it looks like it's more, maybe sticking my neck out here, which you see with tau, more of a general protein stabilization feature. Under certain circumstances, you stabilize a lot of proteins by putting O-glucnag on. Certainly if you look at tau or synuclein, massive differences in stability when you put the sugar on there. So it's looking more like its role in a developed animal is a generic protein stability one, with the link to phosphorylation adding some subtlety there. In development, it's very unclear. I mean, the O-glucnag transferase in development has other roles. It's a polychrome repressor and changes Drosophila development. It was a known mutation in Drosophila. So clearly, during development, it does have rather more specialist roles, but in a developed animal, it is looking a lot more like it's a general protein stability thing. But that reciprocal link to phosphorylation does impact on a lot of different biochemical events. some things have been well dissected. Yes, the RNA polymerase loading is very sensitive to the Yoel state, for example, but there's not that many, to my mind, people in the field might disagree with me, clear biological examples of the role of Yoel beyond its link, its reciprocity with phosphorylation.
I suppose the follow-up is that, many of the kinases really have very specific interaction partners. That's why you have, there's so many different types, they target on specific groups. So in your case, how is the specificity achieved?
Well, there doesn't, on the hydrolase, I mean, we're doing some work now on various glycosylated peptides to look at specificity. There is some, but it's not great. I mean, clearly, kinetically, some substrates are faster than others, but that's ongoing work. The transferase, with its huge array of 13, 14 TPR repeats, clearly there is a lot of specificity hidden in there somewhere, and how it has some protein partners impacting on the glycosylation of certain substrates. But that's just a huge area to be looked at in the future. So.
Debbie?
I was wondering if you could comment a little bit about the mechanism of the heparinase inhibitors, because I'm interested in your matrigel migration assay, and it seems hard to believe that it's just a physical barrier and that you're preventing degradation of it, but maybe I misunderstood.
Yeah, no, I think in that assay that the matrix is the physical barrier, and it's degradation of that matrix by heparinase that allows the cell migration. That's the basis of the assay. It's to assay the effect of heparinase degrading the matrix to allow the cells to pass through.
Even though there's so much other material
that's making up. Yeah, but if you can degrade the heparin sulfate layer, you can allow cell migration, and that's used to do like these initial screens with various inhibitors. If you can inhibit the heparinase, you can prevent the cell migration.
Great. Okay, Gideon, thank you very, very much.